what we can see here is a three-dimensional structure of a protein. Now, the question is, why do we need to know these structures? Well, firstly, because we need to understand the function that these proteins play. And we also, and this is absolutely crucial for drug design. So the question is, well, this particular protein, is not just any protein. This is ubiquitin. And ubiquitin is a very important protein as it forms about up to 2% of all the proteins in our brain. And in particular, it actually participates in this protein recycling process that Konica explained very in great detail. Its role is actually to tag those proteins which are then used which are degraded later. And the reason I'm interested in that protein actually is because it forms a knot. A knot, just like, a knot on any shoelaces sometimes, where there's a kind of knots we actually don't want to have them on our shoelaces. But these knots actually are important in this case because they prevent the ubiquitin from being degraded along the protein that it tags. And so what we are interested in is how to find these knots and to be able to analyze and see what roles they play for proteins. But before we can find them, actually we have to find the structure. And that we have two possibilities. One is via experiments using the latest cryo-electron microscopy, or a more traditional approach where we basically point X-rays at a protein's crystal and then observe the diffraction image. But this method is very accurate, but it's very costly. The other option is to use AI, and this is a total revolution in this field, introduced a couple of years back, and last year awarded the Nobel Prize in Chemistry to the authors of this model. The great achievement is also because this gives us a similar accuracy while lowering the cost substantially. And therefore, as a consequence, whereas we have about 230,000 discovered experimentally 3D structures of proteins, we have over 200 million predicted ones. So once we have the model, the 3D structure, the next step is to apply a mathematical knot theory. And for that, we have actually come up with a library where we worked together with mathematicians and implemented these, they're called polynomial invariants, these kinds of algorithms that are used to detect knots, but also slip knots. Well, a slip knot is actually what we want to have on the shoelaces, right? We want to untie them easily. And then we also have lassos, which are like a cowboy's lasso, basically the same, a similar topology, as they're called. So once we have that, we have this library, we have these algorithms, we can apply that to a huge amount of data, basically AlphaFold's 200 million structures. We took 160 million of them. These are the higher quality ones. And then we came up with a database. We discovered 700,000 knotted ones. And this ended up in a database called AlphaKnot, right now in the version 2.0. And we also looked at the lassos. This is a more recent work, just published 2 weeks ago. And there are almost 16 million such proteins which form lassos in the 3D space. So these databases are free for everyone to use. You can just look them up and have a look at these proteins. They have also some nice visualizations, and every researcher can search through them and can see how these three-dimensional features can influence the further research in drug design, but also material science. Especially the lassos are more important in the case of material science. So thank you very much for your attention, and have fun with today. Thank you.